to be here. So I will talk about statistical issues in the study of stem cell differentiation using transcriptome sequencing, or in short, RNA-Seq. It's been a pleasure to work on this project with my students and postdoc. And in particular, the work that I will present today was done mainly with Fanny Perraudot, David Erisso, and Kelly Street. And I'd also like to thank John Nye, our collaborator on this project, for motivating this work, and also for being a fantastic collaborator for almost 20 years now, and also his group member, Dia Das. She's here in the audience, a BITS fellow, and she'll be my backup for the talk, and his postdoc, Russell Fletcher. And I'd also like to acknowledge my collaborators, Livan Clément in Belgium, Elizabeth Purdom at Berkeley, Jean-Philippe Vert at the Mines School in Paris and Institut Curie, and Nir Yosef here at Berkeley. And this work was funded by a grant from the BRIN Initiative, so I can't help but show this beautiful picture. So here's an outline for my presentation. I'll begin with a historical perspective on the role of statistics in biology. Then I'll present the biological question that motivated our work, and the rest of the talk will be devoted to a survey of the main step in a workflow for the analysis of single-cell RNA-Seq data, with, in particular, exploratory data analysis, normalization, expression quantitation, cluster analysis, and the inference of cell lineages and pseudotimes. And I should say that much of this work is still in progress as we're getting our hands dirty with lots and lots of data and trying to make sense of all of these really interesting data. So there's a long and fruitful history of joint development between statistics and biology, and data has always been at the core of this. A natural starting point is Mendel. Mendel is known as the father of genetics, and his 1866 paper is actually a great read. You can find it on the web. Mendel's laws of heredity were entirely based on statistical inference applied to data from very carefully designed experiments. It's statistical analyses that allowed Mendel to make the bold leap from his experimental results to theoretical conclusions. He reverse engineered the Punnett squared in the sense that he only observed phenotypes or traits of the pea plants, but he inferred the genotypes from these. And now we know that Mendel's hypothetical factors are genes or segments of DNA that code for protein, but the experimental confirmation came much later. His work was ignored for about 50 years, and it took another 100 years or so before we could really make sense of what he saw in his experiments in pea. So he was very careful, again, with experimental design. And this is a diagram that summarizes some of the experiments that he did to study the inheritance of flower color in the pea plant. It's an intercross. He did that for several traits and to study also the joint inheritance of pairs of traits. And the data that he collected are actually quite simple and quite small by today's standard. They fit on a page. They're basically counts for combinations of phenotypes. So here we have different plants, and this is the experiment to study the inheritance of the form of the seed. So it can either have the dominant phenotype brown or the recessive phenotype angular, and we have this 3-to-1 ratio that corresponds to the first law. Next, another unavoidable figure is R.A. Fisher. R.A. Fisher made fundamental contribution to what is statistics today. He made contributions to experimental design, analysis of variance, likelihood inference, sufficiency, linear discriminant analysis, and so on. But he was also grounded in subject matter applications. In particular, he worked in population genetics and in agriculture. Another great read is his 1952 paper, which is still very relevant today. And in this paper, he studies the relationship between statistics and biology. And in particular, he makes the observation that genetics and statistics are both characteristic products of the 20th century. He makes the connection between factorial designs and Mendelian factors. He talks about randomization, experimental design, and also the need for absolute realism and the importance for statisticians to be in direct contact with what's done in the field. So it's a terrific paper that I highly recommend. An interesting footnote to Mendel's work is that it was revisited by Fisher, and in particular, Fisher noted that the counts that were reported by Mendel were actually too close to the counts that were expected under the first and second laws. So here's some of his observations. Closer than would be expected in the best of several thousand repetition. And this other quote is quite funny. I have no doubt that Mendel was deceived by a guarding assistant who knew only too well what his principal expected from each trial made. So now fast forward to today and the reign of omic data. So today's statistical inference questions in biology are truly multivariate. They involve the joint analysis of multiple, diverse, and high-dimensional data sets. In particular, we have data for entire genomes, transcriptomes, epigenomes, proteomes, metagenomes, exposomes, and so on. We have phenotype data, and we also have biological annotation metadata. By metadata, I mean data on data. So this would be data that you find on the web in databases for gene ontology, or gene pathways, protein structure, or literature data from PubMed. Today's data are complex in several respects. They're complex in terms of their dimensionality. We can be in the small and large P paradigm. Like with early microarrays experiment, we had data on thousands of genes, but only 50 or so individuals. We can also have the large N, large P setting with today's single-cell transcriptome sequencing experiments. The data are of multiple types. They can be quantitative, qualitative, text, graph, or image data. The data can be censored, erroneous, or missing. They come at various levels of processing. Like in micro experiments, you can have image data, or base calls, or already summarized expression measures. The data are dynamic and evolving. Just think about DNA sequence databases or literature databases. And they come from multiple sources and location. So we're no longer just with this cute little XN by P matrices. So this is just summarizing the different data types. So we want to understand the relationship between these different data structures, and in particular, how they affect phenotypes or our health, for example. So where does statistics come into play in all of this? Well, hopefully early enough so that statisticians are involved in the design stages of the project. Experimental design is essential to make sure that we can actually answer the question that motivated the study. It's still very common in the literature to find experiments where the biological effects of interest are completely confounded with nuisance technical effects. So for example, if you see a difference in expression measure, you won't be able to conclude that it's due to a difference between cell type. It could just as well be due to difference between batches. Okay, so just a few moments of thinking about experimental design could save a lot of trouble. We also want to answer the question as accurately as possible, given the available resources. And this has to do with sample size notions and variance notions. And then having designed the experiment, we collect data, and then we try to answer the scientific question. And this could take the form of parameter estimates or tests of hypotheses. And we want to provide measures of goodness of these results. And in statistics, these take the form of bias, standard errors, risk, false positive, false negative error rates. So here are some important aspects of biostatistical practice. The first one is essential, and that's perhaps the hardest step. And it has to do with translating the biological question into a statistical question. And in statistics, questions are parameters. So in this context, it's essential to think about the meaning of probabilities. What are the sources of randomness? What are the sampling units? So for example, it wouldn't make sense to do a t-test when we have an entire population. Are we sampling genes, or are we sampling patients? And also, we want to make sure that the method actually answers the question. If you look at the literature, again, you see a lot of methods, sophisticated methods that are applied without having really asked a proper question. So you're not quite sure what you're answering at the end of the day. Having a question, then comes the time of designing the experiment, then collecting the data. And then it's also essential to look at data to avoid garbage in, garbage out problems. And histograms and pie charts are not the only answers for looking at data. There are many other summaries of data available. And finally, we answered the question that motivated the study. So this could take the form, again, of parameter estimates or hypothesis tests, or sometimes just simple, sensible data summaries. Software is essential. It's needed every step of the way. And BIDS members actually have made key contribution to the area of statistical computing. I'm thinking in particular of the Jupyter project, which is led by Fernando Perez. Another key aspect of research is computational reproducibility, and it should actually be a sine qua non condition to publication, just part of good scientific practice. And here I'm referring to a very narrow notion of reproducibility, just being able to reproduce the computational results of a paper. And that's to be distinguished from the much more ambitious notion of biological reproducibility, whether the findings from one study hold in another. Many tools are available to generate reproducible document, but it's still far from the norm in the literature. So just wrapping up this introduction, the development and application of sound statistical methodology involve constant feedback between subject matter, inference, and computation. I believe that interdisciplinary problems are best addressed by teams of experts in the component disciplines that speak a common language. In that respect, I think the term computational biologist is quite vague because it could be applied to a statistician like me that's working on the analysis of biological data, or one of my biology colleagues that's using statistical and computational methods. Okay, so now let's move to the actual biological question that motivated our work. This is part of a collaboration with the NILAB. And the NILAB was interested in studying the differentiation of stem cells in the mouse olfactory epithelium. And we're focusing here on horizontal basal cells, or HBCs. They're adult tissue stem cells. And it turns out that the p63 protein promotes self-renewals of HBCs by blocking differentiation. So you can think of this p63 protein as a switch that decides between two alternative fates, self-renewal or differentiation. And to study differentiation of these HBCs, we applied single-cell RNA-Seq to cells that were derived from wild-type mice and also mice that had the p63 gene knocked out. And we looked at 5 time points following tamoxifen treatment to get better time resolution for the differentiation process. So here's a diagram of the mouse head, and the epithelium is located right here. These are the different cell types of the epithelium. We have HBCs here in green, GBCs, global basal cells, in blue, then olfactory receptor neurons, subtentacular cells. And we're interested in studying how these horizontal basal cells can self-renew or differentiate into subtentacular cells or neurons. And we're using single-cell transcriptome sequencing. So the first step in a single-cell RNA-Seq experiment is to isolate individual cells and then extract RNA from these cells. And we did this using the Fluidigm C1 system. The RNA molecules are then, if you want, this is my cartoon of RNA-Seq. So the RNA molecules are just chopped up into pieces and then sequenced. So at the end of the sequencing process, what we have are millions of reads, and these reads are just sequences of 100 or so bases, ACGTs. And we then map back these short reads to the transcriptome. And the idea behind RNA-Seq is that when you count the number of reads that map to a particular gene, it gives you a measure of the expression level of that gene. Okay, so after various pre-processing steps like image processing, base calling, and sequence alignment, the data structure that we have are these huge matrices that have cells as rows and genes as columns, and the entries of the matrices are read counts. Okay, so we have discrete read count data. So this diagram here summarizes the design of our olfactory epithelium experiment. It's a bar plot of the number of cells per batch. And here a batch is a C1 run. And the bar plots are color-coded by the biological condition. The biological condition refers to whether the mice were wild type or knockout, and the time point after tamoxifen treatment. So right away, we can see that there's an issue with the design, and that's the fact that batch effects, those are technical effects, unwanted effects, are nested within the biological effects of interest. So here within this orange condition, we have 3 batches. So this is something we'll have to be aware of when we continue the analysis. Yes?
Quick question, perhaps naive for a non-biologist. I assume you're looking at RNA instead of DNA because you want to look at gene expression.
Gene expression, transcription, yeah.
So if that's true, why not just look at the distribution of the ultimate proteins or whatever would be encoded by the RNA in the cell?
So that would address a different question downstream, which would be protein level, and it would require a completely different type of assay. And I think it would be valuable on its own. And again, I'm not a biologist, so maybe a biologist might want to jump in and add further to that. But it would be a totally different type of assays. These sort of microarrays or sequencing assays were great for DNA or RNA, nucleic acids. So protein have a totally different structure and would require different types of assays. That's the statistician's answer. If there's a biologist in the room. I see a hand at the back. Dia?
This is the first step.
First step is a biologist. Yeah.
And later, there are people looking at proteins.
Yeah. Thank you. Thanks for the question and the answer. So this is our design. So keep in mind we have nesting. OK, now let's look at data, finally. First, the problem of zero inflation with single-cell RNA-seq data. This is a bar plot of the proportion of genes that have 0 count in each of the cells. So there's 1 bar per cell, and the bars are color-coded by batch. So this is extreme zero inflation. Around 80% of the genes have 0 counts in a cell. OK, so this is common to RNA-seq experiment. The zeros can be due to either biological reason, the gene is simply not expressed, or technical reasons, low capture efficiency. Zeros are a problem right away. For normalization, they're a problem. I'll have common normalization methods like the one implemented in DSeq. Eliminate any gene that has any 0. So they end up basing their normalization on, say, 5 genes out of 20,000 genes. So it's advisable to do some 0 count gene filtering at the start, but also keep in mind that we need to address zero inflation later on. And now let's look at quality control measures for the samples. So these are measures that reflect alignment characteristics of the reads or sequence composition of the reads. These are box plots of a variety of QC measures stratified by batch. So the message here is that these QC measures vary between batches. Some measure may point right away to samples of poor quality that we want to filter out. Another issue is that the QC measures are associated with the read counts, with our expression measures. So this is a scatter plot of the first principal component of the QC measures against the first principal component of the counts. So we have an association. And here are bar plots of the absolute correlation of the count principal components, so principal component 1 through 5, and individual QC measures. So we might want to keep track also of QC measures when we normalize the data. These are box plots of the gene level log counts for each of our samples. So each box plot, they're tiny, very narrow, because we have 600 or so cells in here. So box plot for each cell of the log counts, color-coded by batch. A more informative display is the box plot of the RLE for each gene. The RLE is the relative log expression, so it's a log ratio of read count to median read count across cells. If the cells had similar distributions of read counts, the RLEs would be centered at 0, and they would have, the box plots would have the same width. So this is far from being the case. You have large differences in distribution both within and between batch. And then finally, I'm looking at an old data set to illustrate the importance of looking at data before jumping into black box analyses. This is an old yeast bulk RNA-Seq data set where we're looking at RNA-Seq for 4 yeast species, Saccharomyces cerevisiae, Paradoxus, Micate, and Bianus. So just by looking at these simple box plots of log counts, we noticed 4 very low box plots. We went back to our collaborators and asked them to look at their lab books or their files, and it turns out here that these 4 samples had mislabeled species. So we were mapping back to the wrong genome. Okay, so just getting very poor data. So when we corrected the labels, we got much better distribution of log counts. So really essential to look at data. So this brings me to the next topic, which is normalization. The goal of normalization is to adjust read counts for a variety of gene level and sample level technical effects in order to allow meaningful comparison of the expression measures both within and both between genes and between samples. So gene level effects could be due to length or their GC content. And technical effects for the samples could be due to batch, like C1 run in our case, or the QC measures. Normalization is essential before any downstream analysis like clustering or differential expression analysis to make sure that when we see a difference in expression measure, it's really due to the biology of interest and not nuisance technical effects. And normalization is even more important for single-cell RNA-Seq than for bulk RNA-Seq because of higher levels of technical noise and also the zero inflation. So if we look at the literature, there's a whole bunch of normalization procedures that have been proposed. Most of them are adaptations of what's been proposed for microarrays or bulk RNA-Seq. So a natural question, and a first question is, does it matter? Does normalization make a difference? Actually, it does. It turns out that the choice of normalization method can have a greater impact on the result than the choice of method for differential expression or for clustering. So we really have to make sure we do this right. The second question is which method is best, and that's not so easy to answer. And the best method depends on the data set. Pardon me. So what we need is a data-driven approach to select an appropriate normalization procedure. And this is what motivated our work with scone. And this is a collaboration with Nir Yosef and Elizabeth Purdom. And with scone, we want to provide a general framework for normalization of single-cell RNA-Seq and other data. So Courtney Schiffman, one of my students, for example, is applying the same types of ideas with metabolomic and adaptomic data. So in scone, we implement a variety of normalization procedures that range from simple global scaling method, where you just use a single number per sample to scale the counts, to more complex regression methods based on either known factors of unwanted variation, like QC and batch, or unknown factors of unwanted variation, like we've done in this remove unwanted variation method, RUV. And it also provides performance metrics for the normalization as well as graphical summaries. And it's available through the Bioconnector project. So this is a diagram of the regression model that's used for normalization in scone. So we model the gene level log counts in terms of 3 quantities. So biological factors like cell type, known unwanted factors, this would be like batch or QC measures, and unknown unwanted factors. This is what we would estimate by factor analysis or singular value decomposition. This is the RUV method. And we're also providing a variety of different metrics to assess performance of normalization procedures. The idea is that we want to quantify the trade-off between the ability of a normalization method to remove unwanted variation and the ability to preserve the variation of interest. So there are 3 main classes of metrics. One of them refers to the clustering of samples according to factors of wanted and unwanted variation. The clustering strength is measured by silhouette width. Another class of metrics looks at the association between expression measures and factors of wanted and unwanted variation. And then the third class of metrics looks at the distribution of the relative log expression. So then we can select or rank normalization procedure based on functions of these different metrics. So we applied scone to the olfactory epithelium data set. And in particular, we looked at 172 different methods that involve various combinations of scaling, regression on unknown and known factors of unwanted variation, adjustment for biology, and adjustment for batch. These are two of the best performing methods according to our metrics. They both involve full quantile normalization. So full quantile normalization is basically matching the quantiles of the count distribution across cells. So it's a pretty aggressive form of normalization. And they also involve regression of the gene level log counts on principal components of the QC metric. So here it's regression on the first 4 principal components, and here it's regression on the first 2. And these results actually make sense when we go back and think about our exploratory data analysis plots. And here it's important to note that when we do regression on 4 QC measures, we have to also adjust for biology, and that's probably because if we adjust for 4 as opposed to 2 QC principal component, we end up removing some biology. So by adjusting for biology, we make sure we keep the biological effect in the counts. So this is one of the main plots returned by scone. It's a biplot. Each point on the plot corresponds to one of the 172 normalization methods. The points are color-coded by the mean performance score. So blue corresponds to bad performance, yellow to good performance. So these would be the good performing method, and they tend to score highly in terms of the scone metric for correlation of read counts with factors of wanted variation that we derive based on positive control genes. This is the same plot, the same biplot, but now we've color-coded the methods according to batch and biology adjustment. Okay, here the poor performing methods in blue corresponds to when we do batch adjustment without adjusting for biology. And because of the nesting, when we adjust for batch without adjusting for biology, we remove the biological effects. So that's why nesting is important. Yes?
So by kind of boiling down much of the information you're giving here to my simple brain, what you're doing is you're running a regression. You find some factors that you're interested in, some you aren't, and then you're normalizing to somehow take up the effect of those that you're not interested in. Why is that preferable to simply saying, you know, I have some factors I'm not interested in, and whenever I look at the results, I kind of know that they're there operating? Because I would think that feedback loop of saying I'm not interested in things and I'm going to normalize, to me that would be kind of a scary thing, like that could obscure things, as opposed to keeping explicit. These other things we're not interested in them, but we have to read each observation, taking them into account.
So we don't want to normalize too much, and I think that's one of the things you mentioned, right? So I know batch is not something that's desirable, but I have this confounding problem, so I somehow need to figure out a way for normalizing for batch, but not removing the biology. But I also may be missing unknown unwanted effects, right? In some experiment, I may only have like one technical variable that's measured. I can do that. This is what I do. I don't like batch, but I also have to be aware of the design.
Yeah.
And I don't like QC. Right? But if I normalize for batch and for QC, I may be normalizing too much, right? So that's why I need some way of ranking the method, and that's what we're attempting to do with these metrics. But also, I'm open to the possibility that there may be other factors that are affecting my read counts, and that's why we have this also unknown component to the model. And actually, in some data that we have, we don't have QC measures. So we were kind of stuck basically having some form of unsupervised normalization. So there's certainly the risk of normalizing too much. And that's why we try to have these metrics where we use control genes to evaluate that. I'm not sure if I answered the question.
Largely.
Okay. Thank you. So again, this is where design matters. Nesting here, we have to also adjust for biology. We can't just remove for batch blindly because we're removing the biological effects as well. The next couple plots are just making sure that we're obtaining results that make sense. So this is looking at our box plots of RLEs. If you remember the box plots before of the RLEs, they were all over the place. So here, normalization does what it's expected to do. It stabilizes the box plots across cells. In this experiment, we also had negative control genes. So those are genes that are expected to be constantly expressed throughout the cells. And these are box plots of the RLEs for the negative control genes. They're fairly stable. Also, we've decreased the association of read counts with QC measures. So this is just checking normalization works as expected. One final thing, which hopefully also adds to what we were just discussing here, this is looking at what the unsupervised RUV normalization returns. So this is kind of like a factor analysis that's based on the negative control genes. And this is a plot of the factor returned by this unsupervised normalization. You can see here it's color-coded by batch, so it does capture on its own some of the batch effects. And here, this is a scatter plot of the QC first principle component versus this unsupervised normalization factor, and it also captures some of the QC. So we could have almost ignored that we had batch information and that we had QC information, and then the unsupervised normalization. It would have captured similar effects.
So you're saying it would have captured one of the things you actually had information on?
Yes, exactly.
So that's
they're not. So in case
you can name your factors to some extent.
We're relating. It's reassuring. Yes, exactly. It's reassuring that we can actually relate what this unsupervised normalization does to information we have. So basically, it makes me feel more confident if I get a data set where I don't have any of these batch or QC measures to say I'm going to use the unsupervised normalization. Thank you. OK, so this was normalization. And I always have planned to talk about way too many things. So let me see how far I make it. So we tried to bring together within one framework a lot of the issues that I've mentioned so far. And for this, we've developed a method that's based on a zero-inflated negative binomial model. And we want to do within that framework handle the zero inflation, over dispersion, and experimental design. And in particular, with an appropriate parameterization of the negative binomial mean and zero inflation probability, we can do normalization, dimensional reduction, and differential expression analysis within the same framework. So this is the model that we have for this zero-inflated negative binomial distribution. So the log of the negative binomial mean and the logit function of the zero-inflation probability are modeled in terms of 3 components. So it looks a lot like the scone model, except it generalizes it, right? So we have in green known sample level covariates. In blue, we have known gene level covariates. So the gene level covariates could be length or GC content. And in red, we have unknown sample level covariates. So here, the known sample level covariates could correspond to either variation of interest, like cell type, or unwanted variation, like batch, that I've mentioned earlier. And likewise, the unknown sample level covariates could be of interest. This would be the clustering case where we want to discover novel cell types, or they could correspond to unwanted variation. This would be our unsupervised normalization. So within this framework, we can accommodate different types of experimental designs. In particular, we can handle nesting issues by an appropriate formulation of the design matrix X. We can perform normalization through the X, V, and W matrices. And we can also perform dimensional reduction. This would be the W matrix, and I'll show you an example of that. And we can also do that differential expression analysis, so finding genes that are differentially expressed between different cell types in 2 different ways. The first way is very simple. We just use the zero-inflated negative binomial model to derive posterior probabilities that zeros belong to the count component. And then we use these weights in standard bulk RNA-Seq methods. So this is very simple. A more complex approach is to do likelihood-based tests on either the negative binomial mean or the zero inflation probability. So looking at the negative binomial mean would correspond to looking for a continuum in differential expression between different cell types. Focusing on the zero inflation probability would correspond more to on-off differential expression patterns between the cell types. So again, this ZinB-Wave method has been implemented in a package that's released on Bioconductor. And I'm going to illustrate very quickly the differential expression functionality and the dimensionally reduction. So for that, I'll turn to not the OE data set, but a variety of publicly available data sets. So this is a plot to this figure. We look at dimensionally reduction for a glioblastoma single RNA-Seq data set. The cells are from 5 different patients. And here we're comparing dimensionally reduction based on ZinB, the W matrix, with 2 factors, to dimensionally reduction based on principal component analysis with total count normalization, and zero inflated factor analysis with total count normalization. And for ZinB, we don't need any prior normalization. It's done as part of the model. So these are scatter plots of the first 2 components for each of these 3 dimensionally reduction methods. And the cells, each point is a cell, and the cells are color-coded by patient. So you can see that you get better grouping by patient for our ZinB method. The bar plots below are similar to the bar plots I showed earlier, and they're bar plots of the correlation, absolute correlation between the components for each of the 3 methods and the QC metrics. So we've reduced correlation with the QC metrics with the ZinB dimensionally reduction method. So this is again looking at dimensionally reduction with ZinB, comparing it to zero inflated factor analysis with 4 different normalization methods and PCA with 4 different normalization methods, and now it's with 4 data sets. And we're looking at the strength of the clustering by biological condition. And we measure clustering strength by average silhouette width. So for the first 2 data sets, so this is a cortex data set, this is another cortex data set, and I think hippocampus data set, glioblastoma, and mouse embryonic stem cell data set. So we already had labels for the cells. So we want to check the ability to recover these biological labels with each of the 4, with each of the different dimensional reduction method. So we get, again, better clustering by biological condition with our ZinB method. So this was really encouraging. And now I'm going to turn to the differential expression application of ZinB-Wave. In particular, this is a comparison of various differential expression methods on simulated RNA-Seq data. These are scatter plots of true positive rates versus false discovery proportions. So it's in a simulation setting, so we know what are the genes that are differentially expressed and those that are not. And the 2 curves here at the top that perform best are standard bulk RNA-Seq differential expression method. Those of you that have used bioconductor packages have heard of EDGAR and LIMA. But we've downweighted the zero counts in these methods using weights derived from the zero inflated negative binomial model. And the other curves here correspond to unweighted D methods and also newly proposed method for single-cell RNA-Seq. So we were, again, very encouraged by these results. So just a quick survey of ZinB-Wave. Let me see how much time I have. So the next step here, so let me go back to the OE data sets. So again, we're interested in studying this differentiation of these HBCs into neurons and testicular cell. So before we can look at this directly, we need to identify the states in the differentiation process. So before inferring the lineages, we'll cluster our cells. So this is some of the motivation for the work we've done on cluster analysis. We wanted to have a clustering method that's robust to the choice of samples, that's also robust to the preprocessing and the clustering algorithm. We wanted to avoid focusing on the number of clusters, just because very often the number of clusters is not of primary interest to the biologist, and also because methods for selecting the number of clusters can be very sensitive to the choice of preprocessing method or to cells or other tuning parameters. So the approach we've taken is to use resampling-based sequential ensemble clustering. Ensemble clustering is basically aggregating multiple clusterings that were obtained either from resampled versions of the data set where we apply the same clustering method, or to using the same version of the data but applying different algorithms to it. And it's essentially what's been done already in supervised learning with bagging, boosting, and random forest. So the idea is to improve stability and then bore strength across different methods. So again, we have a bioconductor R package for this RSEC. So these are the main steps in RSEC. And we start with a base clustering algorithm. So it could be k-means or partitioning around medoids, PAM, and associating tuning parameters for the clustering. So if we do clustering on a principal component, it would be a certain number of principal component. It could be the number of clusters. It could be a design matrix. And then from this base clustering algorithm, we generate a single candidate clustering. And then we have the option here of also doing sequential clustering as proposed by Tseng and Wang in 2005, and also doing resample-based clustering, like on bootstrap samples of the cells. So this gives us one candidate. So we repeat this process to generate a collection of candidate clusterings. And then based on this collection of clustering, we identify a single consensus clustering based on the co-clustering matrix. I'll show you examples of that in a moment. This often produces many clusters, so we have a pruning back of the clusters. So we'll be merging non-differential clusters by creating a hierarchy of clusters, working up that tree, and then testing for differential expression between sister node and collapsing nodes that have insufficient differential expression. The standard next step, once we've clustered the cells, is to identify signatures for the clusters. In other words, find genes that are differentially expressed between the different clusters. And it's not trivial. Suppose we have clustering of the cells in 10 different clusters. Sort of a default approach would be to compute F statistics for all the genes. But this is not satisfactory because it's not a very specific test. It's testing for any difference between the clusters, and it's also sensitive to outliers. So instead, we turn to a very standard approach in statistics, which is to use cluster-specific contrast. And by doing this, we can also rely on the machinery of generalized linear models, where we can use all of the samples to test for cluster-specific contrast. So it gives us more precision. So the types of contrast we can look at could be all pairwise comparison between clusters, or just one cluster against all, or something that's a little bit more clever is to use the hierarchy between the cluster and test for differential expression only between sister nodes. So let me show you some of the results.
Do you expect the dendrogram to matter here? There's some notion of hierarchy that I've missed?
For the cells, yeah, there should be. There's the cells, yes, there's a hierarchy between the cells. So in that case, it will make sense to test for for finer differential expression than just like one against all. Hopefully the next figure.
The source difference is evolutionary?
Part of it is evolutionary, and then subtypes of the different cells. Yes, and Dia might want to add to this. Thank you. I'm lucky to have Dia. She should be like just sitting right here, right? She's way at the back. Thank you. Okay, so I'll just show you quickly some results from applying RSEC to our olfactory epithelium data set. So we applied a whole bunch of base clustering algorithms. So this is a plot where rows correspond to clustering algorithms. So like say k-means with different k's, with different numbers of principal components, with different ways to resample the data. And then the columns correspond to the samples, okay? And there's a fixed ordering at the top. What changes is the color labels for the cluster, and we try to maximize the overlap by choosing the colors differently in each row. So you can sort of see that for some samples, there's a pretty stable clustering across methods, but for others, there's a lot of noise. Okay, so we summarize these results in the co-clustering matrix here. So rows and columns correspond to samples, and then the co-clustering matrix is just the proportion of times that a pair of samples are clustered together. So this is how we aggregate the many candidate cluster into one consensus. So when we then do clustering, it's sort of like clustering on the clustering. So we do clustering based on this co-clustering matrix. That gives us many clusters, and then we do pruning by merging non-differential clusters. So this is the sorts of this, I think this is what Dia mentioned. So when we apply RSEC to our OE data set, we got clusters that corresponded to a variety of states in the differentiation process of HBCs. So we recovered the HBCs, but we also identified global spatial cells, microvillus cells, immediate neuronal precursors, immature and mature olfactory sensory neurons, and immature and mature sustentacular cells. Then just quickly back to the cluster signature. So this is one of these standard images you see in genomics. This is a heat map or a pseudocolor image where cells correspond to read counts for genes here and cells here. So this is our clustering of cells. And this is a heat map where we took the genes that had the most significant F statistics, the top 472. And the 472 was chosen because of the next plot that I'll show you. So this is really, we don't really see any meaningful grouping when we do differential expression based on the F statistics. And that should come as no surprise because it's not specific at all. However, if we do this dendrogram-based approach to testing for DE, so we test for DE between sister nodes, we get much more interesting patterns that biologists can inspect now. So what we did here was looking at the hierarchy and testing for DE between pairs of sister nodes. And at each node, we took the 15 most significant DE genes. And that gave us 472. That's why I have 472 in this plot. So much more interesting patterns that biologists can look at now. And I think I just have a couple minutes, so I apologize to Kelly because I'll rush through what he has done. And that's the method for bringing everything together and deriving the lineages and the pseudotimes. So just a very quick mention of that and tell you a little bit about the results. So it's Slingshot. And the goal of the method is to infer cell lineages. And the input to the method, so really, really quickly, is gene expression measures that have been normalized and that have in a space of reduced dimension, and cell clusters. Okay, this is why we take the input from RSEC, sorry, the output from RSEC as the input to Slingshot. And Slingshot returns cell lineages. Those are ordered subsets of cell clusters and cell pseudotimes, as well as weights for each lineage. Our approach to this problem is based on a minimum spanning tree that we apply to cell cluster. This gives us more stability than doing a minimum spanning tree on individual cells. We can semi-supervise the MST by specifying root and leaf nodes. In particular, we can specify leaf nodes when we know what the terminal states are. In our case, we know that terminal states are neuronal cells and sustentacular cells. And how we infer the pseudotimes along these lineages are using an adaptation of the principal curves algorithm of Hastie and Stutzel. And here's quickly the results that we got. So when we apply Slingshot to the RSEC clusters, we identify 3 lineages: HBC to sustentacular cells, HBC to microvillus cells via GVCs, and HBC to olfactory sensory neurons via GVCs. We also looked at differential expression along the lineages, so finding which genes are differentially expressed for a given lineage. We applied generalized additive models, and we found that there were very different processes for the differentiation of HBCs into testicular cell and the differentiation of HBCs into neurons. For testicular cell, the process was via direct conversion of HBCs without cell division. But in contrast, for the neurons, there was the intermediate stage of GBC and much more marked changes in the differentiation. So this is sort of the diagram for what Slingshot does. Dimensional reduction, the MST, and then the smooth principal curves. And this is a heat map of genes that were found to be differentially expressed across the first lineage for the neurons. So I'm sorry to rush through that. Just want to make sure I'm finished on time. So just I'll end with a few concluding remarks. So just stress again the importance of design. Just applying very simple design principle could salvage experiments. Validation, we're desperately in need for controlled genes and samples, for normalization, for differential expression analysis. Computational reproducibility is still far from being the norm, and there's still confusion with biological reproducibility. And then just some important notes to keep in mind when we analyze data. So look at the data. Don't reinvent the wheel. This is from French cartoons, the Shadok from the 60s and 70s. Why do it the easy way when you can do it the hard way? Beware of hammers looking for nails. Beware of models and assumptions. The results should be driven by the data, not by the models. And then when we report probabilities, think about the interpretation of these probabilities. So sorry for rushing through the last part of the talk, and thank you very much for your attention.